Hello everyone, I'm going to walk you through the process of using Microsoft Excel to generate a standard curve from a Bradford assay for the purpose of estimating the protein concentration in an unknown sample like a cell lysate. So any solution that contains mostly proteins, if it doesn't really contain any other sorts of molecules, it should appear as a mostly clear and possibly yellowish or slightly brownish solution as you see here. So the reason why that's important is because proteins, since they're mostly colorless, you're not going to be able to use spectrophotometry to estimate their concentration just on their own unless you're dealing with a solution that contains one particular kind of protein. Now, what you can usually do in those cases is you can do UV spectrophotometry at a wavelength of 260 nanometers, provided that the protein that you're measuring contains lots of tryptophan, tyrosine, and phenylalanine. But if you have something like a cell lysate that contains many, many thousands of different kinds of proteins, that may not be a good option because you can't necessarily be sure that all the proteins in that sample are going to contain those 3 amino acids. So in those cases, you generally want to use an assay like a Bradford assay or like a BCA assay in order to help you estimate the total protein content quickly and cheaply and without any problems. So you would want to do this for future applications like loading your samples onto an SDS page gel because generally you want to know approximately what the total protein concentration of your sample is so that you can load an equal amount of protein into all the wells. So I'm going to present here a general procedure, a general protocol for doing a Bradford assay. There are several different ways you can do this. You can scale it up or scale it down depending on how many samples you want to test. If you have a lot of samples, you may want to do all of this in a 96 or 24 well format. I'm going to present a procedure here that is appropriate for just a very small number of samples, maybe 1 or 2 unknown samples that you want to quantify. So something that you're going to need is you're going to need what's called a standard curve. What a standard curve is you take a solution of protein that has a known concentration, usually a solution of 1 milligram per milliliter bovine serum albumin, and you make a range of concentrations of that bovine serum albumin solution, and then you get the absorbance of those solutions so that you'll have a direct relationship between absorbance and concentration, and that should be a linear relationship. Therefore, for any unknown samples that you have, all you need is to get their absorbance, and then you should be able to figure out what their concentration is from the standard curve. So, to get started, you'll want to get at least 5 microcentrifuge tubes. That's how many you'll need for the standard curve. Plus, you'll need an additional tube for every unknown sample that you want to quantify. So for our purposes here, we're going to pretend we have 2 unknown samples, so we would need a total of 7 microcentrifuge tubes. So tubes 1 through 5, like I said, are going to make up our standard curve. We're going to use our 1 milligram per milliliter bovine serum albumin solution, and we are going to plot a linear relationship between the absorbance of those samples and the concentration of BSA, which we can figure out later on. So we are going to add 0, 1, 2, 5, and 10 microliters of BSA to tubes 1 through 5. So tube 1 would contain no BSA, and that's what we're going to use to blank the spectrophotometer. Tube number 2 would have 1 microliter, tube number 3 would have 2 microliters, and so on. And for each tube, we want to bring the total volume in the tube up to a total volume of 100 microliters with distilled and deionized water. We want every tube to have exactly the same volume in it, but of course the amount of BSA we added is going to vary. Okay, and then for tubes 6 and 7, we are going to add 2 microliters of each of our unknown samples to those respective tubes. And then similarly, we would want to bring the total volume up to 100 microliters with distilled and deionized water. And then finally, you can see here, we want to add 1,000 microliters of Bradford reagent, which is a solution that contains Kumasi brilliant blue, a molecule that will change its absorption pattern when it binds to protein and therefore appear blue, hence the name. We want to add that to each tube and then mix everything together well. We'll need to wait at least 5 minutes to allow this binding to occur, and then we are going to want to put these solutions into visible cuvettes and read their absorbance on a spectrophotometer that is set to 595 nanometers, the absorptive peak for Kumasi Brilliant Blue when it is bound to protein. Okay, so in a second here, I'm going to show you my Microsoft Excel spreadsheet, but basically, like I said, we want to take our collected data. So if you want to imagine that you follow the protocol I presented on the previous slide, and then you read the absorbance of each of your samples, so you'll imagine that you've collected your absorbance values and you have those all ready to enter into Microsoft Excel. So what we're going to want to do here is we want to plot a linear graph in which absorbance at 595 nanometers is on the y-axis and BSA concentration is on the x-axis. So at this point, we would already have the absorbances of all of our samples. The unknown samples are still unknown, but before we can really start to figure those out, we need to quickly calculate what the BSA concentration in each tube was for tubes 1 through 5. Okay, so the original concentration of BSA was 1 milligram per milliliter. That's what we call the stock solution. Or you could also say, to make things easier for us, that would be the same thing as saying 1 microgram per microliter. So the total volume in the cuvette that we placed in the spectrophotometer, you will recall, is 1,100 microliters. So a total of 100 between the BSA and water, and then an extra 1,000 for the Bradford reagent. So if we had added 1 microliter of the stock solution, like we did for tube number 2, the new concentration after this dilution would be 1 microgram per 1,100 microliters, which would be the same as 0.000909 micrograms per microliter for that particular tube. So that would be the BSA concentration in that particular cuvette at the time that we read the absorbance on the spectrophotometer. So now I'm going to show you how to calculate this in Microsoft Excel. Okay, so here's a spreadsheet that I'm looking at here. Let me go ahead and zoom in for your convenience. So what I have listed here, just to kind of organize all of our data, I have our 7 tubes, 1, 2, all the way through 7. You'll recall 1, 2, 3, 4, and 5 are reserved for our BSA solutions. And you can see here that I've indicated that to each tube we have added the indicated amount of BSA. We did not add any samples, so you can see that sample was only added to tubes 6 and 7, and those are currently unknown. You can see how much water we added to each tube to make sure that we end up with a total volume at this point of 100 microliters. And then finally, you can see I've indicated that we add a total of 1,000 microliters of the Bradford reagent to get to a total volume of 1,100 microliters in every tube. Okay, so at this point, we're going to mix everything together. We have all that. Then we're going to read the absorbance of each tube at 595 nanometers, and you can see that I've actually already put that in here. I mostly made these numbers up, but when you do a standard curve, this is pretty much what you can expect to get. You can see a linear increase in absorbance as the concentration increases, but that's what we don't know yet. We don't actually have anything inputted for the BSA concentration here. And like I said, that's something that we need to calculate. So an easy way to do this, you can do what I did on the slide that I just showed you. You can use the dilution formula, or you can just do this. So what I'm going to do here for the first one, and this one should be easy because we should understand that if we didn't put any BSA in there, the concentration should be 0. So what I'm going to do is I'm going to highlight the amount of BSA that we added. If we added no BSA, there should be no micrograms of BSA that we added, and we'll divide that by the total volume of 1,100. And that, like I said, is going to be 0. We'll drag this down to the rest of them, and you can see that for the sample calculation that I did on the previous slide, where I said we add 1 microliter to 1,100, you can see we get the concentration that we had figured out there. So now we actually have our BSA concentration and our absorbances for our standard curve. So now what we want to do is we want to plot that out. So I'm going to highlight those 2 sets of cells. I'm going to insert, a particular kind of graph called a XY scatter plot, and you can actually see the plot that we get here. I'm going to make this bigger so you'll have an easier time seeing it. Okay, so just plotting this on its own is not going to be sufficient for what we want. So we want Microsoft Excel to do what's called a linear regression for us. We basically want it to fit the data, assuming that this is a linear relationship, of which it is. That is a good assumption that we can make. So, and more importantly, I want Microsoft Excel to give us the equation of the line. So what I'm going to do here is I'm going to click on any of the data points, right-click, and I want to add a trend line. So you can see in the menu that's popped up over here on the right, I have an option of fitting the data to a number of different mathematical functions. I want this to be a linear function, which is what it should be. If I try to say that it's logarithmic, polynomial, or anything like that, I'm probably going to get an error because Microsoft Excel is going to know from the data that I've inputted that, hey, dummy, this is linear, you ought to be clicking linear. So I don't want to mess with that. So I'm going to come down here and I want to do two things. I want to click Display Equation on Chart, and then I want to display an R-squared value on the chart. Okay, I'm going to make that a little bit bigger for you. I'm going to make the font bigger so you'll have an easier time seeing that. Okay, so two things here. The R-squared value, which can only be a number between 0 and 1, the closer that number is to 1, the better the fit the data is for the linear function. So having an R-squared value that is very close to 1 is a good thing. It means that your assumption of a linear relationship is a good one, and practically speaking for your laboratory work, it means that your pipetting technique of adding all the reagents into the tubes and getting the absorbances was very good. If you get an R-squared value that is less than probably 0.95, and even that's probably a little low, that's evidence that your pipetting technique was probably very sloppy, so you'll probably want to try this again. So as long as you get an R-squared value that's very close to 1, that should be good enough, and we don't actually need that number beyond that. The equation of the line is what we want here. The equation of the line is given in the format of y equals mx plus b. So y is the value of the y-axis, that is the absorbance of the sample. X is the concentration of the sample. The 43.966 here, the m, that is the slope of the line, and then the last figure here, the 0.0041, that is the y-intercept. We are going to need all of that information. So to make my calculations easier, I am going to put this right here, 43.966 for the slope, and for the y-intercept, I will put 0.00, try to fix this here, 0.0041. I'm going to need that information a little bit later on. Okay, so let me jump back here real quick. So like I said, the first thing we needed to do was calculate the BSA concentration in our first 5 tubes for the purpose of graphing this out in a standard curve. So like I said, we can calculate it this way just by using a calculator and rearranging things to make it that way, or we can actually do it in Microsoft Excel. Either way, you'll get the same number. Okay, so once we plot that BSA standard curve, which we just did, like I said, we need to verify the data are sufficiently linear. Like I said, if you get an R-squared value that is not very good, you should start over because your pipetting was very sloppy. Also, as I said, the equation of the line is what we were after, and Microsoft Excel will give you a rough approximation of what the equation of the line is. Y equals mx plus b, where m is the slope of the line, y is the absorbance of the sample at 595 nanometers, x is the protein concentration in micrograms per microliter or milligrams per milliliter, they should be the same thing, and b is the y-intercept. Okay, so here's where we can start to take steps towards figuring out the concentration of our unknown samples. Because we have the absorbances, the Y, because we have that information for our unknown samples, we can actually plug those into this equation, rearrange the formula to get X all by itself, X being the concentration of those protein samples that we are after. If we solve for X, we should have what we want. Okay, so let's start to figure out how we're going to do some of these calculations. So before we go any further, let me go ahead and jump back to Microsoft Excel here and give you an idea of what we're going to do next. Okay, so first thing we're going to do here is we want to use the equation of the line, y equals mx plus b. So in order to get x all by itself, we need to do a little bit of very simple algebra to rearrange the formula so we get x all by itself. This should take the form of x equals y minus b divided by m. So in order to do that, what I'm going to do is I'm going to come over here to a little offset I have here for tube number 6, and I'm going to do just that. So x equals y for that sample. The absorbance of that sample is 0.25, so I'm going to click that, minus the y-intercept, which I have right here. I want to put those in parentheses because Microsoft Excel will give me an error probably if I don't, and then I'm going to divide all that by the slope of the line. So that tells me that the concentration of tube 6 was 0.005593 micrograms per microliter. I'll go ahead and do the same thing for tube number 7. I want to use the absorbance in tube 7. I don't want to reuse the absorbance for tube 6. So that minus the y-intercept, put it in parentheses, and then divide by the slope again. So the concentration of tube 7 is 0.0071851 micrograms per microliter. Now, what you might be thinking at this point is those concentrations for our unknown samples seem very low. Well, here's the issue. What we just calculated was the concentration of the protein inside the cuvette. Remember, we passed light using the spectrophotometer. We passed light through the cuvette. That cuvette contained 2 microliters of protein from the unknown sample, 98 microliters of water, and 1,000 microliters of Bradford reagent for a total volume of 1,100 microliters. So what we really want to know, what was the concentration of that original protein sample before we diluted things? So that 2 microliters of protein we took from the original tube, we want to know what the concentration was in that original tube. That's not what we just figured out. We just figured out the concentration of protein in the cuvette, the 2 microliters of protein with all the other stuff too. So what we need to do here is we need to use the dilution formula to work our way backwards to find the original protein concentration. C1 V1 equals C2 V2. So the way this is going to work is that C1 equals the original concentration of the protein sample. This is what we want. We don't know it yet, but this is what we're going to be solving for. C2 is the diluted concentration in the cuvette, and this is what we just got done calculating previously. V1 is equal to the 2 microliter volume of the original sample that we added to the cuvette, so that's something that we know as well. And then V2 is the 1,100 microliter total volume inside the cuvette. So you'll notice here, for an algebraic formula that contains 4 unknowns, we already know the value of 3 of them, so we can very easily solve for the 4th one that we do not know. Okay, so let's set up this to see how things are going to go. So let's say we take tube number 6. We calculated the concentration of tube number 6 as being 0.00559. So let's just verify that real quick. Yes, that's what we got right there. Okay, so C1, that's what we're going to solve for, times 2, that's the 2 microliters of how much of it we added, equals 0.00559 micrograms per microliter times the 1,100 total volume inside the cuvette. So what we should be getting here is the original concentration of the sample was 3.08 milligrams per milliliter, or 3.08 micrograms per microliter. So let's do this in Microsoft Excel to verify that that's what we're going to get. Okay, so you can see what we calculated before I have indicated as the diluted samples, so now I've got another set of them that are designated as being the original. So what I'm going to do here is I'm going to say, okay, if we're going to solve for C1, that's going to equal C2 V2 divided by V1. So I'm going to take the original concentration here, I'm going to multiply times 1,100, I can click any one of those, I'm going to put parentheses here again, and then I'm going to divide by 2, which is right here. So 3.076. So you can see when I account for rounding, that's exactly what we get. And then if I do the same thing for tube 7, I'm going to take this concentration here, multiply by 1100, and then divide by 2. For that sample, I get 3.95 micrograms per microliter. So what you'll actually appreciate here is that tube 6 and tube 7 have different protein concentrations. They're not that far off, but they are definitely different. So the idea here is that if you're wanting to load these samples for something like SDS Page and you want to load equal amounts of protein, you're going to have to add slightly different volumes to make sure that the total protein content in the two wells is going to be the same. So for example, if you wanted to add 30 micrograms of protein, then a very easy calculation we could do is you could say, okay, for tube 6, which is 3.07, if I want 30 micrograms, I would have to add 9.75 microliters of that in order to get a total of 30 micrograms. So 9.75 times 3.076 should give you 30. Similarly here, if I want 30 micrograms, I would do the same thing. But one thing you may have to be wary of that would nullify these calculations and make you have to start over is you're probably going to have to add lamellary sample buffer to your original tubes, and that's going to dilute down the concentration. So if you're adding 2x lamellary buffer, that would be a 1-to-1 dilution, and you would just have to cut these concentrations by a factor of 2. If you're adding 4x, then you would have to cut it by a factor of 4, obviously. So that's something to consider. People kind of like to add different concentrations of lamellary, or it depends on what kind of stock concentration they add, but that is something that you have to consider. You're not just going to be adding the lysate directly to the sample because you need that lamellary sample buffer for the SDS page because you need the SDS to denature and coat the proteins and all that good stuff that you're probably already aware of. Okay, so that is going to do it for this little instructional video on how to use Microsoft Excel to plot standard curves for a Bradford assay and how to do the calculations to calculate your protein concentration. I hope you found it helpful. Thanks for watching, and I will see you next time.